Hello, Dr. G. Today, Hui Ing and I, Isaac, will be talking about the advancement of technology through the explanation of CRISPR-Cas9 complex. In today's era, technology has advanced many divisions in the science domain. This technology allows scientists, or more specifically, molecular biologists, to create innovative ideas utilizing organisms such as bacteria and viruses to our advantage. One of the discoveries scientists are trying to unriddle and perfect is the dynamics behind an ongoing research called genome editing. Genome editing is a form of technology used to alter the sequence of an organism's DNA. This directly affects the organism's cellular mechanism and enables the target organism to elucidate the functional organism of the genome at the systems level, as well as identifying causal genetic variations. One newfound technique in genome editing is called CRISPR. CRISPR is an acronym that stands for Clustered, Regularly Interspaced Short Palindromic Repeats, and like its name, it is a repetitive strain of DNA sequences found in the genome of prokaryotes such as the bacteria species Staphylococcus pyogenes. The CRISPR technology employs the use of an enzyme called Cas9 belonging to bacteria and the guide RNA. Together they form a complex known as the CRISPR-Cas9 system, used for the alteration of an organism's DNA. CRISPR-Cas9 was first discovered in the 1980s as just mere repetitions of genomic material in E. coli. However, in 2007, a scientist named Barango and his colleagues uncovered its function through the demonstration of Streptococcus thermophilus acquiring resistance against the bacteriophage by incorporating a fragment of the viral genome into its CRISPR locus. This allows the bacteria to memorize other invading bacteriophages and prevent viral infections by inhibiting the start of the lysogenic life cycle of the virus. The CRISPR-Cas9 system has excited many scientists for its benefits, such as it being reasonably cheaper, faster, more accurate, and more efficient compared to other genome editing technologies. This brings hope to the science community because CRISPR could be a saving grace for humankind, carrying a multi-purpose trait that can be used in treating medical disorders or just a study template for more discoveries to come. Now we're going to talk about the mechanisms of CRISPR. CRISPR, along with other CRISPR-associated genes, function as essential effectors in adaptive immunity for many selected bacteria and archaea, as they play a role in eradicating invading genetic material when a response is triggered in an organism. CRISPR-based immunity consists of having the invading DNA or plasmids being fragmented and integrated into a CRISPR locus, which typically consists of clustered sets of CRISPR-associated genes and the signature CRISPR array. On the other hand, Cas genes are translated into proteins, and most CRISPR arrays are first transcribed as a single RNA before subsequent processing into shorter CRISPR RNAs, which directs the nucleolytic activity of certain Cas enzymes to degrade nucleic acids based on sequence complementarity. Since the genome is modified in this process of spacer acquisition, the offspring inherits the protection. New spaces are usually added at one side of the CRISPR, making the CRISPR a chronological record of the viruses the cell and its ancestors have encountered.
Hello, now I'm going to explain to you about the system of CRISPR-Cas9 technique. To your information, the CRISPR-Cas9 mediated defense process can be divided into three stages. The first stage, adaptation, leads to insertion of new spaces in CRISPR locus. In the second stage, expression, the system gets ready for action by expressing the Cas genes and transcribing the CRISPR into a long precursor CRISPR RNA. The precursor CRISPR RNA is subsequently processed into mature CRISPR RNA by Cas proteins and accessory factors. In the third and last stage, interference, target nucleic acid is recognized and destroyed by the combined action of CRISPR RNA and Cas proteins. CRISPR-Cas9 system consists of three types of systems. The type 1 systems are defined by the presence of the signature protein Cas3, a protein with both helices and DNase domains responsible for degrading the target. There are 6 subtypes of type 1 systems are identified, type 1a through type 1f, that have a variable number of Cas genes apart from Cas1, Cas2, and Cas3. All type 1 systems encode a cascade-like complex. Cascade binds CRISPR RNA and locates the target, and most variants are also responsible for processing the CRISPR RNA. The type 2 CRISPR-Cas system encodes Cas1 and Cas2. The Cas9 signature protein and sometimes a fourth protein. Cas9 assists in adaptation, participates in CRISPR RNA processing, and cleaves the target DNA assisted by CRISPR RNA and an additional RNA called track CRISPR RNA. Type 2 systems have been divided into subtypes 2A and 2B, but recently a third 2C has been suggested. The Type 3 CRISPR-Cas system contains the signature Cas10 with nuclear functions. Most Cas proteins are destined for Type 3A and Type 3B complex, which are similar to cascade. Type 1 and Type 2 systems are known to target DNA, while the Type 3 system target DNA and RNA. The Type 2 system can have been exclusively found in bacteria, while the Type 1 and Type 3 system occurs in both bacteria and archaea.
The CRISPR-Cas9 system achieves its sequence specificity by the special structure and conformation of the Cas9 protein. The Cas9 protein contains a conserved core and a bilobed architecture including adjacent active sites and two nucleic acid binding grooves, a large recognition RAC lobe and a small nucleus NUC lobe that are connected by a helix bridge. RAC determines the Cas9 specific function, whereas the NUC incorporates two nucleus domains, RUVC and HNH, a protospacer adjacent motif PAM interacting domain PI. Under natural conditions, Cas9 is inactive. It is activated when combined with the sgRNA at its RAC lobe. The Cas9 sgRNA complex scans a DNA double strand for rigorous PAMs, then trinucleotide NGG using Watson-Crick pairing between sgRNA and targeted RNA. Once anchored at the proper PAMs, the HNH nuclease domain cleaves the RNA-DNA hybrid, while RUVC cleaves the other strand to form a double-strand break. The double-strand break can be repaired by non-homologous end joining and homologous direct repair mechanism that are endogenous to prokaryotes and eukaryotes. Non-homologous end joining employs DNA ligase 4 to rejoin the broken ends, an operation that can introduce insertion or deletion mutations, whereas homologous directed repairs the double-strand break based on a homologous complementary template and often results in a perfect repair. The error-prone non-homologous end joining has advantages for gene knockout. Homologous directed repair is used for gene replacement and gene knock-in implants.
Next, I'm going to talk about application of Cas9 as genome editing platform. The Cas9 nucleus cleaves DNA via its root C and H nucleus domains, each of which sneaks a DNA strand to generate blunt-end DSBs. Two Cas9 nucleus complex with appropriately spaced target sites can mimic targeted DSBs via cooperative nicks, doubling the length of target recognition without sacrificing cleavage efficiency. Expression plasmid encoding the Cas9 genes and the short sgRNA cassette driven by the U6 RNA polymerase III promoter can be directly translated into cell lines of interest. Purified Cas9 protein and in vitro transcribed sgRNA can be microinjected into fertilized zygotes for rapid generation of transgenic animal models. For somatic genetic modification, high-titer viral vectors encoding CRISPR reagents can be transduced into tissues or cells of interest. Genome-scale functional screening can be facilitated by mass synthesis and delivery of guide RNA libraries. Catalytically, that Cas9 can be converted into a general DNA binding domain and fused to functional effectors such as transcriptional activators or epigenetic enzymes. Cas9 coupled to fluorescein reporters facilitates live imaging of DNA loci for illuminating the dynamics of genome architecture, reconstituting the split fragments of Cas9 via chemical or optical induction of heterodimer domains. System from Arabidopsis confers temporal control of dynamic cellular processes. Let's look into other examples of application CRISPR-Cas9 techniques are following. Designing synthetic genetic circuits in cells using the CRISPR system. Character methods for transformation reaction with high performance by CRISPR-Cas9. Improving muscle function in mice with Duchenne muscular dystrophy with CRISPR. CRISPR in treatment of HIV infection. Resistance against malaria by modification of mosquito DNA using the CRISPR technique. And lastly, modeling cancer through the activation of tumor suppression genes and the iteration of oncogenes.
Arguably, the most important advantages of CRISPR-Cas9 over the other genome editing technologies is its simplicity and efficiency. Since it can be applied directly in embryo, CRISPR-Cas9 reduces the time required to modify target genes compared to gene targeting technologies based on the use of embryonic stem cells. Improved bioinformatic tools to identify the most appropriate sequences to design guide RNAs, and optimization of the experimental conditions enable very robust procedures which guarantee successful introduction of the desired mutation. As for the limitation of CRISPR-Cas9, the molecular mechanism exploited to insert DNA fragments is mediated by DNA repair machinery activated by the double-strand break introduced by Cas9. Since the scope of the DNA repair system is not to integrate DNA fragments in a genome, targeted alleles often carry additional modifications such as deletions, partial or multiple integrations of the targeting vector, and even duplications. Secondary unwanted mutational events at the target locus plague standard ES cell-based projects as well. And researchers have learned how to avoid generating mice carrying passenger mutations. To identify the correct recombination events in ES cells, most laboratories use a combination of positive and negative selection procedures and validation procedures aimed at detecting additional mutations at the target site. When performing the CRISPR-Cas9 procedure directly on embryos, on the other hand, it is impossible to select for the desired event, greatly limiting the possibility to identify the desired allele. Moreover, the mosaicism observed in founder mice generated using the CRISPR-Cas9 approach makes the identification of unwanted genomic modifications at the target site very challenging. Thank you, Dr. G, for giving us this opportunity to research about the CRISPR-Cas9 system, and thank you for watching this video.